All right, so let's go on and cover the cells that were left. First and foremost, we will start with the erythrocytes. Now, the erythrocytes, as I said, they're basically red blood cells. They're biconcave disc. Okay, their main job is to transport the oxygen from lungs to the tissues and bring the carbon dioxide from the tissues to the lungs. You think of the red blood cells as an oxygen delivery truck. Why are they biconcave? They're like a donut shape, right? Without a hole. The advantage for this shape is that it increases the surface area, more oxygen can diffuse, there's increased flexibility, and it can squeeze through capillaries smaller than themselves. Now, why does it not have a nucleus? The mature RBCs eject nucleus, mitochondria, organelles, and this leaves more room for hemoglobin, right? So they have no nucleus, no mitochondria, no organelles. This gives us more space to pack the hemoglobin inside. The energy source is not oxygen since they have no mitochondria. RBCs use glycolysis. 90% of ATP comes from the HMP shunt. 10% produces NADPH. Why? Because this protects the RBC from any oxidative damage to the membranes, right? The lifespan is 120 days. Old RBCs are removed by splenic macrophages. Now we have two things, anisocytosis and poikilocytosis. Anisocytosis is when you have different sizes. This is seen in iron deficiency and mixed anemia. And poikilocytosis is when you have different shapes. So you can see sickle cells, they're sickled basically, and spherocytes look like spherocytes. Reticulocyte is basically the immature RBC. It contains residual RNA. The residual RNA is supposed to like be ejected off till the time it becomes a mature RBC. It appears bluish in color. High retic count means that the bone marrow is responding after bleeding or any hemolysis or blood loss, and the erythropoietin rises, the bone marrow works harder, and the reticulocytes rise. And patient bleeds after trauma. Why? Because the retic count rises, meaning the bone marrow functions normally, right? A chloride shift is very important. How does carbon dioxide travel in blood? It travels as hydrogen carbonate. Inside the RBC, the carbon dioxide adds up with the water to form carbonic anhydrase. Now, not true. The carbonic anhydrase basically converts the carbon dioxide and water into H2CO3. Okay, now this H2CO3 is further cleaved by hydrogen and hydrogen carbonate, two hydrogen and hydrogen carbonate. So what happens is you take carbon dioxide, you take the water, and using the carbonic anhydrase, you make the H2CO3. Once the H2CO3 is made, break it again. You break it again into hydrogen and hydrogen carbonate. This hydrogen carbonate leaves the RBC and chloride enters the RBC. So the hydrogen carbonate is exchanged for chloride, right? So this is the chloride shift. Now, why is the chloride shift important? This chloride shift is also known as the hamburger phenomenon. It's very funny, but that's the real name. What happens in the lungs? Everything happens in the lungs. The hydrogen carbonate returns to the RBC and the chloride leaves. The chloride comes back in the cell. Anyways, the carbon dioxide enters the alveoli, you exhale it, and the chloride enters, right? So, why is hemoglobin important? Hemoglobin does more than carry oxygen, it also buffers the hydrogen. Without hemoglobin, the carbon dioxide would not give you hydrogen, which would give you basically say acidosis, right? Without hemoglobin, all the carbon dioxide will give you hydrogens, which will give you severe acidosis, right? So that is something you should know about the chloride shift. Remember that the membranes contain this fluoride and hydrogen carbonate antiporter, which basically allows the hydrogen carbonate to go out and the chloride to come in and transfer the carbon dioxide from the periphery to the lungs for elimination. Now we look at a bit about at this point, I would prefer that you guys pause the video and read this stuff. Basically, I've explained all of this, but it would be helpful for you guys to stop after I explain a certain cell and just read once and then go on because then it'll stay in your memory. It will retain much longer. We move on to thrombocytes now. Thrombocytes are platelets, they're not cells, they're like fragments of megakaryocytes, right? The main job is to form the plug. So whenever what happens is platelet plug formation after vessel injury. We have a few steps of this platelet plug formation, and let's go through them one by one. Now, the first thing that we want you to see is now whenever an injury happens, the collagen is exposed, right? You can see that. Now, what happens is whenever collagen is exposed, von Willebrand factor binds the collagen. Once the von Willebrand factor binds the collagen, the platelet sticks. Now you can see that the von Willebrand factor is getting all this collagen to stick here, right? Now platelet sticks through the receptor. Now the receptor that we use is GP1B. GP1B, you can remember one B, the initials are similar to I, so initial is 1B. It basically does the initial binding. The platelet adhesion, platelets adhere stick via GP1B receptor to one Willebrand factor collagen, right? And then you go on and see that the platelets get activated. Now, the platelets get activated, they release granules. These granules basically release, they recruit more platelets and increase activation, right? So, these platelets are like this chain reaction where you get more and more and more platelets activating. The platelet activations, the platelets activate and release granule contents like ADP, thromboxane A2, calcium 2 plus, etc. Right, so you have all of these platelets activating and these granules being secreting, right? Now, what happens is the next part is that these platelets aggregate using this special factor called the GP2B3A factor, right? And fibrinogen. So fibrinogen and GP2B and 3A both become part of this big platelet aggregation. And when we're talking about the dense granules, if you remember, the dense granules had calcium, ADP, serotonin, and histamine, right? So ADP recruits more and more platelets. The serotonin of the dense granules causes the vasoconstriction, and the alpha granules, I told you the alpha granules had von Willebrand factor. So the alpha granules contain von Willebrand factor, they contain fibrinogen, fibronectin, and platelet factor 4, right? So they're like protein granules. And then we have something called as the thrombopoietin. Let me just revise this here. We had the platelet granules, they had the dense granules and the alpha granules. The dense granules had calcium, ADP recruits more platelets, serotonin causes vasoconstriction, histamine contributes to inflammation and vasoconstriction. Then we had the von Willebrand factor. The von Willebrand factor was very important in helping the platelets basically adhere together. Here they bind the exposed collagen to the von Willebrand factor binds to exposed collagen, right? And the fibrinogen was important for platelet aggregation and clot formation. Fibronectin aids in platelet adhesion and wound healing, and platelet factor 4 heparin binding protein modulates coagulation and inflammation. Alpha granules are protein-rich granules that support adhesion, aggregation, and coagulation. Now, they had something known as thrombopoietin. Thrombopoietin is mainly produced by liver and this stimulates megakaryocytes to make more and more platelets. Whenever you have low platelets, you get these petechiae or tiny red rods here. Okay? Whenever you have low platelets, you get these petechiae or tiny red rods here. That is something I would mention. Okay, moving on to monocytes. Monocytes are the largest circulating WBC cells. They have a kidney-shaped nucleus, which is basically bean-shaped. They have lots of pale cytoplasm, as you can see here. The cytoplasm is pretty pale. They travel in blood, enter tissue, and become macrophages. Think of the monocytes as a baby macrophage, right? And then you have these macrophages that phagocytose bacteria, cellular debris, and senescent RBCs. So what the macrophages do is macro means large and phage means eater. They basically eat the bacteria, dead cells, and old RBCs. Is antigen presentation, right? What is antigen presentation? They basically express MHC2, MHC class 2 receptors, and these present antigens to CD4 cells. So basically, what the macrophages do is they take this bacteria, they basically eat bacteria, dead cells, and more RBCs. But what they do is they express these MHC class 2 and they present the antigens to the CD4 cells because, as you remember, the CD4 cells were helping to have that attack, right? Now we have the granuloma formation seen in tuberculosis and sarcoidosis. Granuloma is activated macrophages surrounded by lymphocytes, right? Do you remember the Langerhans cells, the epithelial histiocytes, and they're activated by macrophages are activated by interferon gamma, which is basically produced by Th1 cells and natural killer cells. So natural killer cells and Th1 cells produce interferon gamma, and this is basically activating the macrophages. Whenever we have macrophages in liver, we call them Kupffer cells. In the brain, we call them microglia. In the bone, they're osteoclasts. In the skin, they're histiocytes. In the lung, they're alveolar macrophages, right? One more thing, the septic shock connection here, the gram-negative bacteria contain lipopolysaccharide, right? This produces a lipid A. The lipid A binds CD14 on macrophages. Once it binds CD14 on macrophages, there's a massive TNF alpha release, which leads to septic shock. So that is something you must remember. I guess I should stop the video there.